Oh, okay.
Okay. Good evening, everybody. Welcome back to another episode of Apart Oceans. Oceans Apart. It's been a while. Declan has been traveling the world, seeing everything from India to Nebraska,
True.
and I have been stuck in the cold in Sweden.
Also true.
Yeah. So, but yeah, yeah. I don't know. What's on the schedule tonight?
Well, today we're going to talk about genetics and specific genes' influence on metabolism. Uh, because Artem was interested, and I have a presentation, so that's what we're going to do. Yeah, that's what I got.
Well, without further ado, if you're ready to go, then I'm ready to listen and ask questions.
All right, let's start sharing screen. Start sharing. We can see Artem and me. Now we can hopefully just see me. And now we can see this. Can you see the?
Yeah, I can
Okay.
see. Actually, for people listening, the title is "Genetic Influences in Metabolism: A Story" by Declan Morrissey.
Indeed. And if you have questions, I can't see you right now, so you're just gonna have to yell out.
Okay, I will.
Give me just a big Bubba kind of shout.
Yeah.
Okay, so we're gonna start with genes of interest, and we have 4 genes of interest. We're starting with myostatin, then we have MSS51, and then we have PPARGC1 alpha, and then we have PYGM. So this whole thing was for an assignment in my genetics class for exercise science, and we were supposed to do like a 20-minute presentation on this, and we had to like list why we had chosen all the genes we chose, and basically this is just my reasoning. Myostatin's cool because muscle hypertrophy. MSS51 is cool because of changes in mitochondria. And mitochondria are super cool. And then PPARGC1 alpha is also kind of linked to mitochondria. And then PYGM is codes for this little enzyme down here, glycogen phosphorylase, which basically just like stores glycogen and breaks it down. And if you lack the PYGM gene, you have this disease called McArdle's disease, and you can't store glycogen, and you just can't exercise really well without like extreme pain. So I think it's pretty cool.
Okay.
And we're starting off with myostatin here. Hopefully someone out there gets the Family Guy reference. But the story behind this is that there's a kind of bull that I'm pretty sure everyone is aware of a little bit. It's called like a Belgian blue cattle. And it's born without a myostatin gene, and so it kind of looks like this pig does, like it's just huge and jacked. There's been like 1 or 2 humans known in history to have the mutation where they just don't have a myostatin gene, and so it's a muscle growth inhibitor, which is downregulated with resistance training. So basically, what that means is if Artem goes to the gym and lifts heavy weights, then myostatin is decreased in its expression, and so he's able to grow muscle. But my presentation was about metabolism. So metabolically, it preferentially shifts muscle towards type 2 fibers, changing the metabolism of glucose. So if you go back to your basic ex-phys stuff, then type 2 basically uses glycogen and glucose much better than type 1s do. And then high levels are associated with obesity, insulin resistance, and low levels of HDL cholesterol, which takes cholesterol from peripheral cells to the liver, basically like controlling fat metabolism there. So what's cool is obviously insulin resistance. You can't really use insulin to shuttle glucose in, so it changes glucose and fat metabolism, which is kind of interesting too, ya boy. All right, and so then actually having a myostatin deficiency decreases mitochondrial respiration. So again, if you're going to the gym and you're lifting weights or you're that Belgian blue cattle, then you're decreasing mitochondrial respiration, which is interesting. But I found an article that said it might be due to a CoQ4 decrease, which through a malate aspartate shuttle in the heart and a glycerol 3-phosphate shuttle in skeletal muscle allows NADH to give the electron transport chain electrons. The glycerol 3-phosphate shuttle is for FADH, which I failed to mention, but, still probably did well in this presentation. I don't know. And then there's an enzyme called PEPCK in the liver. And when myostatin's not expressed, the liver is more able to store glycogen. So, which is kind of cool because then you're able to release glucose a little better into the bloodstream. So that's kind of cool. Am I right?
I just have one question. Like this, the CoQ4 decrease. Like, what is that? The causation of it. Just
all right. So if we go to this next slide here, we'll answer Artem's question all in one go.
Okay.
So this is a malate aspartate shuttle. Mm-hmm. So basically, you can start it anywhere here, but oxaloacetate is one of the Krebs cycle products. And if you just move through this, right, you have malate dehydrogenase, which basically takes a H from NADH and converts it to malate. Malate is then moved across into the matrix of the mitochondria. And then, you can kind of just keep moving through this whole process over and over again. Where CoQ4 comes into play is by pulling this NADH towards the mitochondria so that it can actually be used to in like this reaction, like the malate dehydrogenase reaction, and to give you the hydrogens for the electron transport chain. So it acts as like an attractor, basically. Like if you're looking at it, like my mouse just froze. Okay, here we go. It sits kind of like down here and pulls this NADH towards itself. Mm-hmm. Does that make sense?
Yeah.
All right, perfect. And then we have a glycerol 3-phosphate shuttle. So if we move back to our XPhys again, at some point during glycolysis, which is right down here, we basically get a product called glycerol 3-phosphate. And what you can do with that is you can basically like shuttle that into the mitochondria to give you eventually to give you hydrogens and to allow this FADH to do its work and give you electrons for the electron transport chain. And the same thing occurs here. Like the CoQ4 is supposed to be like right in here and is pulling this towards itself. Mm-hmm. And then then this is my last slide for myostatin. And basically what we were supposed to do was to have like a future directions for research sort of thing. This is like that. This is that for me, basically. So this is going to get weird. But so resistance training is kind of a stressful phenomenon. And so there's stress imposed on the membrane of the cell. And so then this green protein becomes active, which is, I think, sterol regulatory element binding protein 1, I think is what it's called. And basically, in order to repair the membrane, it liberates fatty acids from the muscle cell and then repairs the cell membrane because the cell membrane is made out of fat. So then if you come back to the myostatin and look at the arrow that kind of moves to the down right, so that gene codes for the actual myostatin, and a low level of myostatin has been found to be linked to a low level of fat cells within the muscle. So my thought process here is that basically like myostatin allows you to be better at activating this SEREBP1 protein to liberate free fatty acids. So I think personally, this is like where the research needs to be done, is that the liberated free fatty acids are floating and they move to either this kind of like this protein to be used to repair the cell membrane, or they can be used to repair the actual muscle tissue through arachidonic acid. So arachidonic acid uses free fatty acids as its substrate to repair muscle. And so then by using the already liberated free fatty acids, you would be able to activate the arachidonic acid pathway and cause muscle growth. And arachidonic acid basically just like, there's a correlation between its activation and like muscle cell growth in a nutshell. Any questions there?
No questions.
Fantastic. And I'm fairly sure that I discovered this. I don't think it's like talked about in the literature, so that's kind of cool. If you're out there and you have a PhD and you want to research this, you just mentioned my name. All right, so now we're going to move on to MSS51. So MSS51 expression is downregulated when myostatin is inhibited. So they both kind of work in concert. So if myostatin is inhibited by the lifting of the weights, then MSS51 is also downregulated. So basically when this thing's inhibited, there's an increased level of ATP, beta oxidation, glycolysis, and oxidative phosphorylation. So basically like all energy systems become more active, which is kind of cool. And then fat in mice is significantly lower when MSS51 is knocked out, and the knockout of MSS51 also improves insulin sensitivity in mice, which is weird because, like thinking back to the myostatin, like an increase in myostatin causes insulin resistance. So it's kind of like the opposite end of the spectrum, which is kind of cool, I think. And then, so it's also related to muscle force in mice, but I kind of think that's due to myostatin inhibition and the mice just being like more muscular because the myostatin is inhibited. And then that's all I really had on it because.
Just like a quick question on that.
Yeah, go.
If you go back, like that. Yeah, like with the whole science thing, is it well like is there anything like any research on humans on this? And
No.
like if there's not, like you think, like would you do you have like a proposition like how you maybe would go about it if you would do it, or maybe that's like a bit like way out.
So it's like it's kind of on this slide too. Like I think so. The thing is like how genetics research works is basically like if you want to know the physiology of what's happening, you study it in mice, and like you start with like animal trials. So like they've obviously already started with animal trials for MSS51, but eventually like they should move to human trials. And so like I think as long as it's like deemed safe, basically. And so I think like the biggest thing is probably for humans, I think, is the insulin sensitivity thing, just because there's so many people with diabetes. If there's a potential genetic influence on the diabetes, then that's then you could change that and it'll be for the better. On the same token, like you could probably just have people exercise and see if myostatin's inhibited by their exercise protocol, and then see if this is inhibited by the exercise protocol, and then that is kind of like a genetic influence on insulin sensitivity. So like that would probably be the easiest study design, as opposed to like going in and trying to like alter this gene in some way in a human being. Does that make sense?
Yeah, perfect sense.
All right. So then PPARGCIA alpha, which I'm just not going to keep saying over and over and over again, but it controls for metabolism of fats and mitochondrial metabolism. The expression of this gene increases complete fatty acid oxidation, results in more storage of glycogen. If it's downregulated, it results in insulin resistance. It encodes for PGC1 alpha, which basically is like the regulator for mitochondrial biogenesis. It's kind of the signal for mitochondria to create more mitochondria. And then when the gene is expressed, time of exhaustion is increased. So basically, like it allows you to be more better at using substrates, because the fatty acids are completely oxidized as a result, and instead of like partially oxidized. and you can store more glycogen, which is obviously just going to make it better.
Mm-hmm.
And so what's also cool is voluntary exercise duration is increased when the gene is expressed. And so then it allows exercise to be done at a faster speed with fats as a main fuel source. So basically they measured this on a VO2 max test and with RER. So like as the people were running, they were running faster than when they had previously done the test. And they were like the RER showed they were using more fat instead of carb, which is kind of cool. So it kind of saves glycogen and like glycogen sparing. So yeah. And then, so there's higher enzyme concentrations, and you can just read these, but the ubiquinol cytochrome C reductase and isocitrate dehydrogenase both have to do. So this ubiquinol is one of the electron transport chain complexes, and this isocitrate dehydrogenase is one of the actual enzymes in the Krebs cycle. So that's kind of the fatty acid oxidation process right there. And then the fatty acid to the two fatty acid enzymes and the medium long and very long dehydrogenases are all to do with basically like shuttling fatty acids around in the cell and like changing the different compositions of how long, like depending on how long the chains of fatty acids are. So basically, like you just become more efficient at using fatty acids, and like mitochondria become more efficient, basically. And then what's also really cool is this superoxide, thioredoxin, and catalase all increase, and they have to do with decreasing the amount of free radicals within the tissue. And basically free radicals are like really inflammatory and can kind of really screw you over if you're super inflamed. And they kind of cause like a decrease in how well you function, because they actually alter DNA. They basically like oxidize the DNA, and it basically like deactivate it within that cell, and so the cell dies and can't what's it called, like split anymore. And what else can I say about that? So, and that then these three enzymes basically are called free radical scavengers, where they basically just like pick up the free radicals and then they stabilize them essentially. So it's kind of cool. So you just become like kind of a savage when this thing comes active. And so then what's also cool is a higher expression in the brain prevents toxicity through tryptophan. So tryptophan is a neurotransmitter that builds up or can build up. And it's basically like you can either, you can make basically, what's it called, melatonin out of that. Or I don't think it's dopamine, but there's another neurotransmitter you can make through tryptophan. And the tryptophan recycling basically becomes enhanced when you are expressing this PPARGCI alpha gene to a high level. And then there are two more enzymes that are expressed, the kynurenine and alpha ketoglutarate, which regulate adipose tissue energy homeostasis. So basically, like you just become jacked and lean on this thing. And then, like we were talking about before, the proteins that contribute to the malate aspartate shuttle increase in skeletal muscle. So that process becomes more efficient too. And so again, this is one of the things where like there was very little research done in humans, and it's really, really conclusive in rats and animal models. There were like a few studies I found that were in humans, but a lot of them like were not fantastic. And then just some stuff that could be looked at, like effects of glycogen stores, like the voluntary exercise, VO2 max and peak, and enzyme activity. I really like the length of voluntary exercise one, because I think if if I was like overweight and hated exercise and didn't want to do it, and they said, hey, like you have this gene that like actually you actually like get a really big effect from exercise if you do it. It's this PPARGCIA alpha gene, and you get it from like the 23andMe kit or whatever it is, like, which is like a gene testing thing. You just like swab your cheek, send it to them, and they just like give you your database. And so yeah, like it's kind of cool. And in a nutshell, I think if people like knew they had this gene, they'd be like, oh, that's fantastic, and then like you could target them for specific exercise modalities. So, and let's see here, next one, and then this is the final gene, PYGM, glycogen phosphorylase gene. And so, I kind of talked about this already. Like, there's a little glycogen background here, and it catalyzes the first reaction in the breakdown of muscle glycogen. And so this PYGM gene, there we go, is completely absent in people with McArdle's disease. And again, they can't store or use glycogen in muscle. And then we had to do something about like the interaction of exercise and all these genes. And like I said, McArdle's patients can't really exercise. They can get like this second wind where like if they just like suffer on this treadmill for like, I think it's like 5 to 6 minutes, then they can like exercise a little bit. But generally they like can't. And so like the thing that I think is really cool is PYGM is expressed in astrocytes, which are kind of cells in the brain that help the neuron function, and they turn lactate into a short-term energy source. And then PYGM has been linked to schizophrenia. So if you're deficient in PYGM in the dorsolateral prefrontal cortex, then the deficiency has been found at a high level in deceased schizophrenics, which is kind of cool. And I'll go into like the dorsolateral prefrontal cortex in a second. But so this is kind of just a little graphic demonstrating how the PYGM gene influences astrocytes. So this here, like this is the glycogen storage, which would be completely absent if you don't have the PYGM gene in the brain. So you really like won't be able to give glucose to glycolysis, basically, to make lactate, and lactate then can be shuttled through the MCTs, and it can be oxidized to pyruvate, which can be then dumped into the mitochondria. So that's kind of how that influences the brain metabolism. And the brain usually uses the brain really relies on glucose in a nutshell. Like it's super dependent on glucose. It uses like 20% of our of all our glucose during the day. And then so the future directions, I think more schizophrenia research would be cool. And I think like checking out the hippocampus would be cool, and for this gene and the decision making. So the prefrontal cortex is really pivotal in decision making. And kind of like how you almost like see the world. And so I think doing more research in that in like even non-clinical populations like athletes or general population would be really, really cool. And then overexpression of PYGM, that would also be cool to see if you can just like store a shit ton of glycogen, and then you can just like you can like lift like 90% of your 1RM for like 20, and just like, and it's like 600 or something like that, just something dumb. But that's what I have. Thank you for listening to the 3 loyal fans. Does anyone have any questions?
Well, the only one I can ask questions right now is me, man. But like just off the bat, if you had to pick like one of these genes, like which one would you say is where like if you let's say you could pick one that you could conduct any kind of research you would want to.
Yeah.
Which one would you pick, and then like what kind of research would you want to do?
This was one of the questions I was asked for the class. Like the professor asked me this question. I'm going to exit out of this presentation. Also, just like quickly, these are my references, and if you want me to send them to you,
you could send them. Like we could put it up so if people want to check out.
Yeah,
everything.
Yeah, I also wrote a paper on this, and it's more in depth for sure. How do I get back to Skype? This, and then I do this. So now I should just be full screen again because I'm hey. So I think it's like a really hard question because like part of me wants to do myostatin research because it's just be like, hey, you know what I mean? But the other part of me is like, I think it would be really cool to do research with the PYGM and the brain, and McArdle's, like just to get people to not have such a terrible disease. Like most of them die before age 20, I want to say. So they don't have a super long lifespan, but in terms of genetic disease, it's like it's a pretty decent lifespan for them to have. And then, yeah, like I think PYGM would be the research I want to do, and I'd really like to look at it in the brain. I think that'd be the coolest.
But like with the MacArthur's disease, because I have no idea about it, but is there anything cure towards it? Like if someone's diagnosed with, like from birth or something, is there any cases where like some people kind of it's like gone or?
So there's no treatment for it, but there are things that help it. So creatine supplementation actually helps it, and exercise, even though it's extremely painful for them, does help it a little bit.
What kind of exercise then? Is it aerobic? Or
they can't do anaerobic.
So like just okay, so basically a strength training basically.
Like they can like walk. That's like their biggest exercise.
Yeah. Okay. Gotcha.
Yeah, that's like where it's at.
I'm trying to think if I have any more questions.
I will say about the genetic diseases, like most, like it's cool to say, oh, someone doesn't have myostatin gene, and like they, and they're going to be fucking huge. Like that's great, but most of the time, like people die with these diseases. Like if your genome isn't like a pretty normal genome within like a pretty strict variation, then like you're gonna die.
Yeah.
So, and it's like kind of cool because, I'm gonna just draw out this real quick. There are two kinds of things that go into a gene. One's called an intron and the other's called an extron. And well, that was bad. One's an intron, one's an exon. There we go. And in a nutshell, I'm just gonna explain what that is real quick here. And if we're looking at the myostatin gene. So this is myostatin. Can you see that at all? All right. So basically right here, like there's a promoter region, and you're helping me review for the final for genetics, by the way, right now. So I thank you for that.
I'm glad.
So the promoter region is kind of at the front, and the stop codon is at the back. So basically, promoter, you can it like enhances the expression of the gene. The stop codon stops the gene from expressing. Then the spaces in between these squares are introns. And what's kind of cool is, so like the first space is intron 1, the first rectangle thing is exon 1. And so literally all you'd have to do is delete intron 1 and myostatin's no longer active, because that's the region that like really codes for myostatin. And then you're just like kind of screwed. So it's like a delicate process, and mutations are like cool to talk about. But it's like not, you're like really not effing around when you've got different mutations that like cause some serious stuff.
Yeah. But I was gonna ask, And from the whole research and everything, like, does it say anything about supplementation? Like, well, you mentioned creatine, like
Mm-hmm.
that thing, but like, is there anything else that's like, that you like, they recommend people to take, like any kind of supplementation at all, or just like creatine, and then you can't really do much about anything else?
For McArdle's, yeah, that's pretty much like what it is.
Yeah, but I mean,
Like,
I feel like maybe for other ones, like say, like if you're patient or something, like is there anything that you can, do? Or it's kind of like just right now we just like there's not enough research, you know, done yet to prove anything.
yeah, like I haven't looked into it to be honest, but there's literally like basically nothing when it
Mm-hmm.
comes to like nutrition and genetics that like I think it's like you'd be really hard pressed to find anything on genetics and supplementation.
Yeah.
And it's like there's a bunch of different kinds of there's like, I just have the book right here, so I'm just gonna genetics for exercise science people, and there's like a bunch of different mutations that like I don't really remember right now to be honest, but they like they basically, well, that's not the chapter I want, but there's a lot of different kinds of mutations that can occur, and they all like just like, oh fuck me, where is it? They all kind of like mess you up in different ways, and it just like depends on kind of like almost what happens, because like sometimes the gene, if you're okay, here it is. So like basically, like for example, so if you like, this table is what I'm looking at right now, and I know you like probably can't see that very well at all, but basically like there's a bunch of different kinds of variations, and so sometimes, right, there's a functional effect to all the variations, and sometimes there's just like um, sometimes the protein doesn't act as well as it could.
Mm-hmm.
Like some, right, like I don't know, for example, like GLUT4 like just like wouldn't act like as amazingly as it could if there was a genetic variant. Or sometimes like um, there's like there's just like a total change in the gene sequence, and then there's no coding for GLUT4, and you just don't have a GLUT4 protein. And so and that changes like how well you uptake glucose. And so like there's like you can have like very severe effects, or you can have very minimal effects.
Yeah.
And so like it just kind of depends on what you're looking at.
Mm. Jesus.
Yep. It's been happening a lot the past like 3 days.
It was the same for me, man. Like, it's brutal. Okay, this is like a very general question, man. But did you find the task of choosing the genes hard, and like writing the paper and researching? Like, was it like like kind of just like maybe like real quick, or if you want to take a bit of your time, like kind of how did you
Yeah.
go up? That did you focus on like specific sources, or like kind of like, how did you go about it?
I'm gonna do this. I'm gonna show everyone how to do this, because I think genes are hella cool. And so let me see. I'm gonna start sharing my screen again, so we can see your beautiful face in full view. All right. So you can see that I'm on NCBI and
Yeah.
children training and environmental physiology. All right. So this is free for everyone to use. And so if you just come here and let's say like what's something you're interested in, like anything with exercise science.
We'll use type stress.
Like yeah, so like stress and genetics.
Yeah.
Like that. You know what I mean?
Yeah.
And so, right, we could do some stress physiology. And so let's just go to PubMed. So we have over 100,000 articles. This is a very stressful thing. Let's see. Okay, so let's see. I'm trying to find. So I think, let's see, you know what, let's do this, because like stress, right? So, oh,
Okay.
that was poor. So like we know that's like blood pressure, right, can change with stress. And so, you're looking at, so let's see here. All right. So loss of endothelial endoglin promotes high output heart failure through peripheral arteriovenous shunting driven by VEGF signaling. Okay. So I really have no idea what the hell any of that means for the most part, but VEGF is vascular endothelial growth factor 1. So then, right, like, but we have this right here, this sentence right here. So if I could just highlight all of that. Okay, so then, right, endoglin is a co-receptor for BMP9/10, which is strongly expressed in endothelial cells. Mutations in ENG lead to inherited vascular disorder, heredity, hemorrhagic telin. I don't even know what the fuck that is. But so now we have a gene, right? We have ENG. So ENG is the gene. And so if we just go up here again and we go to, where is she at? And then we just go to gene, and then we type in ENG, hit it, endoglin, right? So this is what we're looking at. Click on that. Now we have our official symbol, our official full name. We have a primary source, gene type, protein coding. So this gene will code for that receptor. It's in just humans, which is great. There's other names for it. And then basically this is going to tell us what it does. So it codes for a homodimeric transmembrane protein. Dimeric is basically just like there's two different parts of the membrane or the receptor, and they're the same. It's a component of transforming growth factor beta receptor complex and binds to beta 1 and beta 3 peptides with high affinity. Mutations in this gene cause heredity, that weird thing. Basically like the vascular dysplasia, so several types of cancer. So it has like cancer influences and all this stuff, right? And so then literally it gives you everything. So it tells you what chromosome it's on. It's telling you the loci, which is basically like where it is within the actual chromosome itself. And then we scroll down here. And then further loci, like more specific, right? And then we have where it's actually expressed. So, the highest, right? So the heart is probably the highest because of this error bar, but
Yeah.
so like, right, we could say, but it's expressed in the adrenal gland, right? And so, like, if we were just going to use your original example of stress, right, we could just kind of, like, this is the wonderful thing about genes is you could just do, like, anything with it. So, like, just a quick, right, so now we have a gene, and so we have ENG, and we kind of know a little more about it now.
Yeah.
And if we go back to PubMed, let's just see if there's anything about it in the adrenal glands. So, right, something popped up about adrenal tumors. I don't know if I can get the full free text, but, we can try this. I don't even know. This is a lot of pressure you're putting on me in a very short amount of time, Artie. All right, so then just going to find it. I don't know if it's there. What?
I think it's the article kind of thing.
ENG. And let's see if it, okay, or it's going to not let me find things. Come on. Oh, no. That's no good. All right, so that was a bust. But then we just come back.
It's part of the process.
Yeah, and so, like, if we just, it's kind of the same. Oh, but like, and I mean, sometimes, like, it is a bust, and,
yeah.
But, again, like, we can just go here, ENG gene and the heart. Boom, right? Like you have, so now, right, you have a systemic review of genetic mutations of pulmonary arterial hypertension.
Mhm.
So, right, an outcome of stress could be arterial hypertension within the lungs, or like around the heart, depending on if they're referring to the actual pulmonary artery or not. And then basically you have this article stating like this could happen because of stress, right? And then you have this gene. And I think, and you can like,
What happened?
so I dumps out on you every 10 seconds. Artem remembers. It's a good time. All right, let's see. All right. So, like ER stress signaling the pathogenesis of non-alcoholic fatty liver disease. Yeah, that's cool, but it's not like really cool. So this like might be something. Determinants of food choice. Like I would bet there's something in here about. There's something in here about genes, right? Similar articles. Nutrigenomics in the modern era. Nutrigenomics is basically like how what you eat, influences your genes. And so, right, then you can kind of come through this and see if there's anything interesting. Maybe read it, maybe don't. Lessons in dietary biomarkers, right? Then you can just kind of come here. This is a twin study, so basically it's looking directly at like if there is a genetic influence of dietary biomarkers. So if you feed a twin the same thing, are they going to react the same?
Yeah.
And so right down here, 60% of microbiome diversity is due to the environment itself. So like, what you're eating, where you're living, are you exercising, are you not exercising, who you spend your time with, what you do for a job, like all that sort of stuff. And then, but then there's like 40% of, there's 40% gene influence there. So you could go microbiome genetics or something like that. I don't know. And then I'm just going to assume that this is a gene. I have no idea what that is, to be honest.
Dude, I feel like we've just deep tracked. Like, I don't know where the fuck we went now.
Yeah, I don't know where we're at either. Humans, just do that.
Well, this honestly proves my point about. Well, actually, not my point, but because I was thinking that maybe like you had some like more like a specific way of doing it, but yeah, dude, like it's a complicated process. Trying finding articles, man. Like it's just because especially like when you have to buy it for like articles and you don't get the abstract and it's like finding the right one, like finding other ones like agree with this statement that's saying here. It's a shit show, man. But,
yeah, it's hard to do, but
yeah, it's fun.
Yeah, it was like, it was relatively easy to just start with like, if I was just like, all right, like oxidative metabolism and genetics and just like researching that.
Yeah.
And I don't love PubMed for like the articles, but I do like Google Scholar is your best friend.
Yeah, no, honestly, Google Scholar is pretty good. I like that as well, man.
So.
Pretty decent.
Yeah, I think that might be a great place to stop.
Yeah, I have no more questions, man.
That's great. I don't even know what just happened for the last like 5 minutes of me just like scrolling through PubMed trying to find genes.
Well, you proved a good point on like people thinking that they can kind of shortcut from finding articles. There is no shortcut. You have to do the work.
Exactly, but all
Yeah,
right,
man.
this has been episode
I think it's section number 14 now.
all right, episode 14 with Artie, Bruce, and Sav, and Declan
Yeah.
and Morrissey with 2 R's and 2 S's.
Ooh.
Thank you for tuning in. Hi, Dad.
Actually, also, hi, Abby. She wanted us to say hi to her, so
Oh,
she's
okay.
now as well, through the walls.
Oh, okay. Whatever.
Yeah.
That's where I'm at.
Yeah. But yeah, thank you to our beloved followers, our 2 beautiful, beautiful followers. And to you returning, I salute you all. And to you new ones, I don't salute you. I welcome you to the family. And yeah, it's good to be back. It's good to see Declan's beautiful face.